Welcome to today's lecture. Today's topic is chemistry of meat, fats or lipids. As we all know, meat has got fat and which is a very important component, and it has got lot of role in as a food, nutrient and in the biological functions. And moreover, there are a lot of controversy about the meat fats which is limiting the consumption of red meat. So here today we will discuss in details about the chemistry aspect. First we will discuss the basic aspects of fat and its chemistry, and then in more details we will discuss about the meat fats, its kind of fatty acid composition and details about the fat content in different species and some of the other properties of fat. So classification of lipids. So before I go for it, let me explain. Lipids are the esters of organic acids and alcohols, and they are insoluble in water, and the salts of lipids behaves like a soap which are very easily soluble and used for cleaning of anything and also for bath. So about classification, basically there are four kind of lipids: simple lipids, compound lipids, derived lipids and miscellaneous category. We will see each of them in more details in the subsequent. So this particular lecture is more suitable for postgraduate students, so that's why I am trying to tell it in more details about the basic aspect before going to the actual meat fats. So here is about the simple lipids. The simple lipids are actually the esters of fatty acids with various alcohols. So normal aliphatic fatty acids and different kind of alcohols, when they react they form esters, and that is called lipids. So they can be two category. One is neutral lipid, that is the ester of fatty acid with glycerol. Glycerol is an alcohol which has got 3 carboxyl group. So each of them can have a fatty acid ester. So such fatty acids are called as fats, are called as neutral fats. Whereas there is another category which are called waxes. So the neutral fats are the true fats or true lipids, whereas waxes are not the true lipids. So waxes are the esters of fatty acid with monohydroxy aliphatic alcohols. So here the glycerol is not there. The fatty acid with aliphatic alcohol makes the esters, and they are of different category like true waxes, cholesterol esters, vitamin E esters and vitamin D esters. All these things are coming under the wax category. The another category of lipid are compound lipids, where the fatty acid with alcohol ester is there plus some other groups are there. Then it is called compound lipids. Like for example phospholipids, where there is phosphoric acid is making the ester with the alcohol plus it has got additional nitrogen base. In the second example is glycerolipids or glycolipids. The examples are cerebrosides, where the carbohydrates are there in addition to nitrogen base but not phosphoric acid. Then another category is sulfolipids, where the sulfur containing acids will be there. And the last category is lipoproteins, where the lipids additionally will have proteins, so they are called lipoproteins. So all these are compound lipids. The third category of fats or lipids are the derived lipids. These are obtained from hydrolysis of simple and compound lipids like saturated fatty acids or unsaturated fatty acids, or mono and diglyceride. That is, in the glycerol there can be 3 ester, sometime one is broken away, so it can be diglycerides, or 2 are broken away, so that is monoglyceride. So they are all coming under derived lipids, or other alcohols sometime coming into it. And there is a fourth category, that is miscellaneous lipids, where we can get aliphatic hydrocarbons or carotenoids, squalene or vitamin E and K. All these things are also coming under lipid as miscellaneous category. Now we have discussed four different class of lipids, in which one of the example is phospholipids I mentioned. So they are polar in nature and cheap cellular lipids and phosphorus containing. So as we know, in the cell membrane there is the phospholipid bilayer that is very important for all biological functions of the cell. So that is having the phospholipid. So phospholipid is extracellular membrane which has got very important function. Particularly in the brain, it contains very high amount of phospholipids, almost 35%, and in the muscles it has got 2% on dry weight basis, whereas in every cell membrane there is phospholipid. Now the phospholipid itself of different category, it is classified into, one is glycerophospholipids or glycerophosphatides. So there is one example like phosphatidyl choline, which is having a common name of lecithin. There is phosphatidyl ethanolamine, which has got a common name of cephalin. These two are most commonly available in the egg yolk. Egg yolk is rich in lecithin and cephalin, and they have got excellent emulsifying properties, for which the egg yolk is used for many food processing as a functional agent. And the third category, which has got attachment to phosphatidyl group, some other constituents like nitrogenous substances, serine or threonine, these are amino acids, or some carbohydrates like inositol, glycerol. So this is the glycerophosphatides or glycerophospholipids. Here is another category of phospholipid, that is the phosphosphingosides or sphingomyelins, and the third category is non-phosphorylated lipids like cerebrosides, sulfolipids, gangliosides, proteolipids, steroids like cholesterol and all is coming in this category. Here just quickly we will mention about the isolation or separation of lipids from the tissue or extraction of lipids from the tissue. This is a practical aspect. For estimation of lipid we need to extract. So the classical method is chloroform and methanol at 2:1. This is a solvent system which is commonly used, established by Folch et al. In 1957. And there are other alternative options like ethanol and ether at 3:1 ratio. Since lipids contain unsaturated fatty acid like monounsaturated or polyunsaturated fatty acid, we are going to discuss more details later, which are easily oxidized or polymerized or isomerized. So because of this issue, we need to do the extraction of lipids at a low temperature and in an inert atmosphere. To prevent this oxidation or other reaction, we need to use low temperature and inert atmosphere for extraction or isolation of lipids from the tissues or from the meat. So in continuation about the separation of lipids, for actual estimation different techniques are used, and one of the most common technique is chromatographic procedures, and which has got different applications like thin layer chromatography, the most classical method, and then the modern method of gas liquid chromatography, and further more accurate method is high pressure liquid chromatography or HPLC, and further we can use column chromatography using silica gel. So these are some of the application aspect applied for estimation of lipids. This should be discussed separately in different lectures. So this is about physical and chemical properties. It's a very separate topic. It can be a separate lecture exclusively. I have discussed about it, some of them in the milk properties related or in the rendering the properties of fat. So here quickly I will mention, first is the solubility, that is how it dissolves. So mostly it will dissolve in the Organic solvent, it is insoluble in water. Then specific gravity, it is about the density in relation to water or relative density. In case of fat, it is less than 1, generally 0.1, 0.91, 0.93. Melting point, that is normally it will be solid most of the fat. When it is liquid, then it is called oil. Fats and oil, that is the difference. Altogether they are called as lipid. So generally meat fats are fat, not lipid or oil. The melting point is about at what temperature it becomes liquid, from solid to liquid. Similarly, there is polymorphism, that is the same compound can have different structural difference, like trans isomer, cis isomer. Boiling point, that is at what temperature it starts boiling. So it depends on the composition of the fatty acid, short chain or long chain, monounsaturated or polyunsaturated or totally saturated. That makes the difference. So generally it can be up to 130 degree, 150, 170 degree. Color, there is a difference due to many factors. Refractive index is another important property for fats. Similarly, the chemical properties like saponification value, that is how much of sodium hydroxide or potassium hydroxide is required in terms of grams for saponifying a particular amount, like 100 gram of fat or 1 gram of fat, that is identified as saponification value. Similarly, iodine value indicates about the unsaturation. So how much unsaturation is there, that is identified by reacting with iodine. If the iodine value is more, that means it is more unsaturated. Similarly, there is acetyl number, there is acid number, Richard Missed value, RM value, that is also related to the quality and properties of fat, and another is Polensky value. These two are more relevant to the milk fat, which we have discussed in earlier lectures. Now, rancidity of fat is a very important aspect. Here the fat undergoes oxidation, either due to the presence of unsaturated fatty acid, it undergoes oxidation, or there will be hydrolysis. The fatty acid is broken from the alcohol and the ester link is broken. In case of true fat, it is glycerides or triglycerides. So that glycerol ester bond is broken. So then there is a hydrolysis and a lot of free fatty acid will be produced. That gives a bad smell and odor. That is very unpleasant. So these are oxidative or hydrolytic. Two kind of changes happens and both leads to development of bad odor and leading to spoilage. So this is very important related to storage of fat, and also in case of meat, because meat has got a lot of fat and that fat undergoes these changes and it affects the shelf life or storage life. Now during hydrolysis, sometimes glycerides further is broken down to peroxides or aldehydes, and this process is influenced by light. So light act as a catalyst in case of causing oxidation or hydrolysis particularly. Similarly, it is faster in refined fats. So refined fats undergoes many chemical process that makes it more susceptible to oxidation. Whereas the natural fats exhibit an induction period of variable duration. So normal fat which is present in meat of different kind, it undergo some resistance of oxidation due to the presence of natural antioxidants. So in our biological system there are different kind of antioxidant mechanism. Many of them give some defense against the oxidation in a normal condition before it starts oxidation or rancidity. Now briefly we will understand about the biological function of lipid. So lipid is coming through every food being used by us, and in meat fat also there is same thing, but in case of meat there are a lot of propaganda against fat. In fact, in all food the limitation for fat has become a big propaganda. So most of the time now we are made to believe that we should not eat fats and oils, we should avoid totally from the food, but that is not always true. because lipid or fat has got a lot of important biological functions as a nutrient and also physiological function. So first one is concentrated source of calories. So lipid is a very good source of calorie, and it is recommended that up to 30% of the energy we can get from the fats or lipids. Similarly, it supplies essential fatty acids and phospholipids. So some of the fatty acids are must for us to consume through food because we cannot produce. So they are called essential fatty acids. Similarly, there are some phospholipids naturally coming for food. So as I mentioned earlier, these phospholipids play important role in the cellular function in cell membrane. Similarly, essential fatty acid has got a lot of important biological function. So it is not wise to remove totally fat from our diet. Then it carries soluble vitamins. Many vitamins are fat soluble, A, D, E, K. They are coming through lipids when we are consuming any fat containing food. Then it spares protein. So if lipid is there, that serves as the source of energy as a second step after the carbohydrate. But if there is no lipid, then proteins will be broken for energy. Then the body's essential requirement of proteins will not be fulfilled. We daily need some amount of protein which has to be supplied from food. If the food is not having sufficient energy, then proteins will be used for energy purpose. Similarly, the fats plays important role for palatability. A food without fat will not have that much good taste. It has important role in growth. As I told, in the cell membrane, lipids are having bilayer, which is very important. So for the growth, there is cell division for which the lipids should be there. Then it provides satiety values in meats. So in case of meat, when we eat, it gives a satiety value, a sense of satisfaction, a sense of fullness comes from that, and it increases working capacity. If we take some amount of fat, we get more energy, the working capacity is better. Now we will talk about fatty acids. So generally we can find them as free fatty acid when they are broken from the linkage of ester with the alcohol. And these fatty acids can be two types, that is saturated and unsaturated. The saturated fatty acid, like most common and major amount of fatty acids in meat and other milk, also milk fat, it is palmitic acid and stearic acid. These are the most common saturated fatty acid found in meat and milk. And then there is unsaturated. So saturated means when there is no double bond between the carbon atoms, whereas unsaturated means when there is one or more double bonds between the carbon atom in the fatty acid chain. Now in the unsaturated, there are several categories like monoethanoic or monounsaturated, dianoic, where two double bond, trianoic, three double bond, polyanoic, there is many. Similarly, it may have branched chain fatty acid, hydroxy acids or cyclic acids. Now, in the second part of this lecture, we are going to focus more on the meat lipids. So before we discuss about the meat lipids, let us briefly understand about the composition of meat, that is the proximate composition or chemical composition of meat, or sometimes we can say nutritive value. In this, the major part is water. We have discussed all these details earlier. The moisture, that is the major part. Water, it is around 70 to 75% in general in different species of meat, and protein is around 19 to 22% in different species of meat, and the fat is around 3 to 7%. This is in case of lean meat. Lean meat means the visible fat, that is the subcutaneous fat, and the depot fat inside are removed. So that's why the lean muscle alone, if we consider, it is 3 to 7%. There is ash which has got mineral around 1 to 1.5%. So this is a rough idea about the fat, how much fat is present in lean meat, but as a whole this fat can be 10, 15, 20%. Now we will talk about characteristics of meat fats or meat lipids. About 18 to 30% of carcasses weight of market steers is the fat. Similarly, in case of hogs, around 12 to 20% of the carcass weight is the fat. So these animal fats are found in three different forms. First one is intracellular, that is within the cell. Second is intercellular, that is between the cells. And the third is extracellular, that is outside the cell. Now briefly we will discuss about the intracellular fats. So these fats are in the form of small droplets in the cellular plasma. Inside the cell plasma there are droplets of fat, and they are mostly consists of unsaturated fatty acids rather than adipose tissue. The considerable amount of this fat could be phospholipids and some amount of unsaponifiable compounds like cholesterol. So the intracellular fats, mostly unsaturated fatty acid, phospholipids and cholesterol. Now the next is extracellular fat, that is the outside the cell. These are located in the network of connective tissue between muscle fibers, and that gives the meat marbling. So marbling is actually the extracellular fat. In other words, we can call intracellular, intramuscular fat, that is within the muscle but between the fibers. So this intramuscular fat or the marbling plays important role in case of beef meat quality. This fat consists true fat about 90% made of straight chain carboxylic acids. Many fatty acids present in beef and pork, which we have discussed earlier. The saturated fatty acid most commonly are palmitic and stearic acid, and the unsaturated fatty acids are sometimes palmitoleic but more commonly oleic acid and linoleic acid. These are the most common saturated and unsaturated fatty acid in meat, but this quantity will vary depending on the feeding regime. So kind of feed given has a role to some extent to modify the fatty acid profile in fat. In case of cattle, the saturated fatty acid is quite higher, even more than sheep and next to pigs. So the cattle has got more saturated fatty acid, and that is one of the concern for related to human health or cardiac health disorders. Now we will focus in more details about the meat lipids. As we have seen, in case of meat, the major component is the water, and then protein, and next comes the fat, which is around 5 to 6% in lean meat, and a very little amount of carbohydrate. Now this water content is variable in different regions, in different species. So whenever the water varies, the fat content also proportionately changes. Rather, it is inversely proportional. When the water content is more, the lipid content will be less. When the water content is less, the lipid content will be more. So these lipids are water insoluble biomolecules that can be extracted from cells and tissues by organic solvents like alcohol, chloroform, benzene, ether. So this we have discussed at the beginning. Several solvents can be used for extracting the lipids for isolation and separation. To discuss further details about the meat lipids, that is the animal lipids are mainly neutral lipids and phospholipids. So earlier we have discussed neutral lipids, that is the triglycerides. The glycerol molecule is estering with the fatty acids, and other is phospholipids where phosphoric acid is the component. The neutral lipids are mostly triglycerides. Sometimes they can be diglycerides also or monoglycerides also. The saturated and monounsaturated fatty acids predominate in meat lipids. So in case of meat, major part is saturated or monounsaturated, but it varies with different species. In case of beef meat, the saturated is more. Even in case of mutton or chevon, also monounsaturated is there, but saturated fatty acid is more. The major saturated fatty acids are palmitic acid and stearic acid. The palmitic acid, that is having C16, that is 16 number of carbon chain, and the stearic acid, that is 18 number of carbon chain. In continuation about the fatty acids, the common unsaturated fatty acids in meat are oleic, linoleic and linolenic. All three are considered as essential fatty acids. That is, these fatty acids must be supplied through our food for our physiological requirement. The oleic acid is the single double bond of the stearic acid, that is the C18, and linoleic acid is the single double bond of the 20 carbon chain fatty acid, and linolenic is the two double bond in 20 carbon fatty acid. So all these are considered as essential fatty acid. In case of pork and organ meats are relatively good sources of linoleic and linolenic acid. So pork and other organ meats are rich in these unsaturated or essential fatty acids. Phospholipids are present in small percentage, that is 0.5 to 1%. They cause discoloration and off flavor in meat products. So these phospholipids are more susceptible to oxidation. There is a unsaturated bond, double bond in case of phospholipids, and that undergoes oxidation leading to discoloration and off flavor. The cholesterol is a minor but important component in meat, and that is always creating a cause of concern. And another is the organ meats, particularly liver, that is also a good source of cholesterol. So this cholesterol present in meat is sometimes giving the negative impact for meat consumption, but this I am going to discuss in other way at the last part. Further, in continuation about the fat content of meat and the fatty acids, the fat supplies a rich source of energy and essential nutrients, as I have mentioned already about the essential fatty acids, and also it is the good source of fat soluble vitamins, that is vitamin A, D, E and K. Generally they are carried through fat. Now fatty acid composition of meat, further if we see, it depends on the species, particularly whether it is ruminant or non-ruminant. Generally, in case of ruminant, the saturated fatty acid is more. The lean and red meat contain similar proportions of monounsaturated saturated fatty acid. However, exact proportions vary depending on type. Lean meat is relatively higher in polyunsaturated fatty acid and lower in saturated fatty acid compared to untrimmed meat. So here we are talking about the lean meat where much of the fat is removed, particularly the surface fat and other internal visible fat is removed. That is the lean meat. So in case of lean meat, the saturated fatty acid is comparatively less, and that is good for health. In a common understanding so far, the saturated fatty acid may cause some health disorder, particularly cardiovascular disease. But it has been observed that the surface fat, subcutaneous fat deposit, that is mostly having more saturated fatty acid. So when we do the trimming, much of this fat from the surface is removed. So the saturated fatty acid content is reduced in case of lean meat. So here once again we will see the different fatty acids. First is saturated fatty acid in meat. The most common is palmitic acid, and the next is stearic acid. Palmitic acid is 16 carbon and stearic acid is 18 carbon, and these two fatty acids are having no connection with cholesterol or health issues. Whereas there is another two minor saturated fatty acid, that is myristic acid and lauric acid. Myristic acid is carbon number 14 and lauric acid is carbon number 12. So generally it has been found that this can increase the cholesterol level more potentially than C16, that is the palmitic acid. So this carbon number 14 and 12 can have more effect on increasing the cholesterol. In continuation about the meat lipids and their nature, so next one most common is mono unsaturated fatty acids. Most commonly we say MUFA, and here the lean beef has similar proportions of saturated fatty acid and mono unsaturated fatty acid. So in case of beef, if it is lean, that is the surface fat is removed and the depot fat is removed, then the lean muscle will have almost similar amount of saturated fatty acid and MUFA. The main MUFA present in meat is the oleic acid, that is the single double bond of carbon number 18, that is the stearic acid. When it has one double bond, then it is called oleic acid. About 30 to 40% of fat in meat is composed of MUFA, that is monounsaturated fatty acids. Now we will discuss about the polyunsaturated fatty acids, or sometimes called as PUFA. Nowadays it is a very commonly known by just all the health conscious people. So this source of essential fatty acids, the polyunsaturated fatty acid includes the linoleic, that is the N6, and linolenic N3. They are all the essential fatty acid I have mentioned earlier. Now intake of N6 is not a problem. Mostly we get this N6. If we eat any food with fat, it will have N6 fatty acid, but intake of N3 is the challenge. Commonly we say omega-3 fatty acid, that is the alpha linoleic acid, that is always limited in most of the plant origin food. So ingestion of this omega-3 fatty acid, that is the long chain polyunsaturated fatty acid, is a big challenge, and sometimes they are called as EPA and DHA, that is eicosapentaenoic pentanoic acid and docosahexaenoic acid. It has been found that increased intake of long chain PUFAs, especially this omega-3 fatty acid, can reduce the atherosclerosis, heart attack, cancer, and all such diseases. So that is why there is a commonly aware to the health concerned people that omega-3 fatty acid should be taken more. Now this omega-3 fatty acid is quite rich in all fishes, and especially the fat rich fishes. So those who take fish, especially fat rich fish, they will never have this issue of limitation of omega-3, whereas those who don't take fish, for them meat is a very good source of this N3, that is the essential fatty acid for better health. Here a little bit more about the PUFAs in meat. So these polyunsaturated fatty acid content to some extent depends on the feeding practices. So if the beef animal are kept on grass feeding throughout the year, then it will have a higher proportion of PUFA. So there is a more demand for the grass fed beef in Western countries. Similarly, in Europe, they have taken up some special project to increase the essential fatty acid or long chain unsaturated fatty acids under a project called Prof beef. So here they have specially tried with some research to improve the content of this PUFA for better human health without compromising the appearance, shelf life, and eating quality. Here we can see the comparison of fatty acid profile of different kind of meat. In case of bacon, the top one, we can see that good amount of saturated fatty acid, and the monounsaturated MUFA is even little more than that. And then there is N6, quite a good amount, and the N3 is very little. In case of pork, the second line, we can see that saturated fatty acid is little less than bacon, and the MUFA is also slightly less, whereas N6 is quite more and N3 is very less. In case of lamb, we can see the saturated fatty acid is quite higher. In fact, it is highest among all, and the MUFA is quite good amount, N6 is quite less, and N3 is very less. And the last line we can see the beef. There also we can see good amount of saturated fatty acid, almost equal amount of mufa, which I have mentioned earlier, almost same, and the N3 is little less and N6 is less and N3 is very less in case of beef. So this gives an idea about the composition of fatty acid in 100 gram of different red meat. Here we will see the distribution of PUFA, polyunsaturated fatty acids, in different species meat and also cholesterol. So in the first column in cholesterol, if we see mg per 100 gram, the mutton, beef and pork, it is quite at the 60 to 80 mg and quite low. Only in brain it is very high, I mentioned earlier, and other organs, that is the kidney and liver, is high in cholesterol. Organ meats are high in cholesterol. Then if we see the C18 is to 2, C18 is to 3 and C20 is to 3. These are the linoleic, linolenic and arachidonic. These are the polyunsaturated fatty acid. If we see in case of 18-2, it is little higher only in kidney and liver, whereas other meat it is somewhat okay. In case of C18-3 also we can see that in case of mutton, beef, pork at a low level, but it is a good source which is normally not available from other food source or plant origin foods. Similarly in C23 we can see some amount of, that is the arachidonic acid, from the brain, kidney and liver. So these are the good source for arachidonic acid. And then there are other long chain polyunsaturated fatty acids. They are to some extent good in liver only. Here we can see the variation between species in a common sense, mostly in case of fat from aquatic animals. The saturated fat is less. The fat coming from aquatic animals, the saturated fat is less, and the presence of long chain unsaturated fatty acid or PUFA, that is C20 and C22, are more. In case of higher land animals, the animals of big size in the land area, the common fatty acids are palmitic 25 to 30% and oleic 40 to 50%. So I mentioned earlier these are the two common saturated fatty acid. However, the common unsaturated fatty acids are palmitoleic and another of course palmitic and stearic is the saturated and palmitoleic and oleic is the unsaturated. Similarly, in case of milk fat, generally we have short chain fatty acids. They are having lot of saturated fatty acid, but more are short chain fatty acid, and also we have palmitic, stearic and oleic in milk fat. Now little bit we will talk about trans fats, that is the transformation of fatty acids which happens due to many reasons, especially due to processing or high temperature treatment. So first of all, the industrial hydrogenation of vegetable oils produce semi-solid and solid fat, which is widely used in food manufacture, in bakeries, in pastries. So hydrogenation is a process to break the unsaturation. So all the oils are having mostly unsaturated fatty acid. So by the process of hydrogenation, in both sides a hydrogen atom is added and the unsaturated becomes saturated, and then it becomes solid, and these fats are used in bakeries, particularly the dalda, hydrogenated vegetable oil, is commonly used, but that is no a concern for our animal origin fat. Sometimes it is found that in ruminants, due to hydrogenation, some cis fatty acid get converted to trans fat. Now, why there is so much of concern is these trans fatty acids are not good for health and cause of lot of disturbances in human health. But these trans fatty acids are mostly coming from the processed food, especially from bakery origin. Similarly, if these fats are used in meat processing, in cooking and frying, that time of course we can get trans fatty acids. So it has been observed in a report in UK that intake of trans fatty acids is quite below the threshold level, that is around 25% of the 2% of the dietary energy as recommended by a standard authority. So here we will discuss some of the modern work or efforts to reduce the fat content and modify the fatty acid composition through modifications and research. So in case of processing, advances in food processing technologies, breeding programs and changes in animal feeds and modern butchery techniques have led to a reduction in the fat content of carcass meat. So by changing the processing of food, so we can reduce the added fat, or in breeding programs there is some change in the animals which will deposit less fat, or changing the feeding pattern that can reduce, or by butchery techniques where all visible fats are removed and we market only lean meat. So all this thing has been successful to reduce the fat content. Similarly, in UK, fat content has been drastically reduced, and more than 30% for pork, 15% for beef and 10% for lamb over last 15 years. So by all these application, they could reduce the fat content in meat because fat has become a cause of concern for human health. And modification of diets, that is the feeding of the particularly ruminants or in pigs, we can change the fat deposition. So cereal based vegetable and fish oil, cereal based vegetable and fish oil, if we are feeding in, it can change the fat deposition or composition, particularly in case of non-ruminants. Whereas in case of ruminants, it is not very easy to change the fatty acid profile of meat because the bacteria, the rumen bacteria plays a major role in fat synthesis. So only by feeding it cannot be interfered much. Here we will discuss about the cholesterol and its connection with human health. As such, conventionally, it is known that cholesterol is connected to saturated fatty acid. So if we eat more red meat, it has more saturated fatty acid, and that can cause more cholesterol and leading to cardiac diseases or cardiovascular diseases. But in 2015, the American Dietary Guidelines Advisory Committee DGAC has made a different recommendation. So earlier recommendation, they have mentioned that dietary cholesterol should be limited to 300 milligram. But in 2015, they have found that the American Heart Association and American College of Cardiology has proved that the dietary cholesterol is not directly responsible for the serum cholesterol. In other words, if we take more cholesterol through diet, it need not increase the cholesterol in the blood. That is why this recommendation has been changed, that there is no need for restriction of dietary cholesterol. So cholesterol is not a nutrient of concern for overconsumption. So if we take this in the right spirit, that means the red meat is not really a cause of concern, especially when we take it in moderation. So those Western countries where red meat is taken in too much, sometimes per capita daily average is 500 gram. There are many people who takes red meat daily 500 gram or more. For them, maybe it could be a cause of concern for many other relevant reasons. But for Indian situation where we may take even once in a week 100 gram or 200 gram red meat, it may not cause any concern. So that's why the all concern related to fat in meat is truly not really required. So here we are at the end of today's lecture. Here let me summarize. Today we have discussed about the meat lipids, the details of chemistry and the relation with human health briefly. So we have discussed what is lipid, different classification and their chemical aspect, characteristics and properties briefly, and different kind of fatty acids. And then in details we have discussed about the meat lipids, what are the common meat lipids, and their fatty acid profile, saturated and unsaturated, the fat content, the differences due to species and how to reduce it. We have discussed about the modern practices and approaches for reducing the fat content in meat and meat products, and we have tried to connect the connection between the meat fat and human health, and perhaps it is not really always true that red meat is the cause for concern for human health or cardiac diseases. So with this I conclude. Hope it is very useful for learning. Thank you very much.